Antigens. Just overview. Innate immune system. To recap. Immune system, it goes with innate and adaptive. Innate, non-specific, it contains cellular component and humoral component. Cellular component, you know up to now. Neutrophil, macrophages and NK cells. Humoral component, cytokines, complements, and other chemokines. Adaptive immune system, later on cell-mediated B cell and T cells, humoral, mainly antibodies. To that, we require antigen knowledge. With some definition, immunogen is an immunogen is any antigen that is capable of inducing humoral and no cell-mediated immune response. But only an immunogen can evoke an immune response. Antigen. A toxin or other foreign substance which induce an immune response in the body, especially the production of antibodies. Peptide. It's a small molecule that elicits an immune response only when attached to a large carrier such as proteins. Carrier molecules includes in body albumin, serum globulin. Right? What is epitope or antigenic determinant? The part of antigen that is recognized by the immune system, specifically by antibodies, B cells or T cells. The epitope is the specific piece of antigen to which an antibody binds. The part of antibody that binds to the epitope is called paratope. Antibody, we'll learn later. So factors influencing immunogenicity, contribution of the immunogen. Foreignness, more the non-self immunogenicity is much more higher. Size, molecular size more than 10,000, 100,000 kilodalton, immunogenicity is high. Chemical composition and complexity. Primary structure, you know, secondary structure, tertiary structure, quaternary structure, or a protein. Once it more complex, so complexity is more, immunogenicity is more. Primary structure we term sequential determinant and others conformational determinant because secondary, tertiary, and quaternary structure has a 3D structures, they are in the conformation. Physical form. If it is particulate, it's much more tend to immunogen. Denatured compared to native, high. Degradability. Antigen processing by antigen presenting cell. Factors influencing immunogenicity contribution to the biological system. Genetics. Now it's about the host, species, individual, responders versus non-responders. Some antigens may evoke high immunogenicity on humans, some may not. Age, also another factor. In naive ones and elderly ones, depends. And also dose, also route of administration. Subcutaneous is the more, then intravenous, intragastric. Adjuvant, substances that enhance an immune response to an antigen particularly. So we use in the vaccination, adjuvants we use like alum. Chemical nature of the immunology. Proteins are much more immunogenic compared to polysaccharides, nucleic acids, lipids. It's a descending order. There are antigens called T cell dependent and independent. Polysaccharides, you know, pneumococcal polysaccharides, lipopolysaccharides, and flagella, they are T cell independent. T cell dependent, there are proteins. Structure likewise, microbial proteins, non-self-altered self proteins, they are T cell dependent. Bit of haploid carrier conjugate. You know structure definition, you know native determinants, heptenic determinants. Likewise, there are heptanes, carrier, no, conjugate. It's a carrier. It's a heptane determinant. Recognized by B cells and antibodies, antigen determinant. I said earlier, composition, sequence determinants, conformational determinants like size, all matters. Superantigens, a class of antigen that causes non-specific activation of T cells, result in polyclonal T cell activation and massive cytokine release. So in the usual process, once antigens is there, normal one, they activate specifically several T cell families only, monoclonal or polyclonal T cell response. Once in 10 to the power 4, 10 to the power 5. But with the super antigen, they leads to polyclonal T cell activation. That's what problem. It leads to massive inflammatory response, immune stroke like strong. Examples there are bacterial, viruses, tubercular, there are but staphylococcus enterotoxin, toxic shock syndrome toxin, exfoliative toxin, pyogenic exotoxins, likewise there are. So later on, determinants recognized by the immune response, innate, adaptive immune system, discrete determinants like tertiary quaternary structure react with specific pathogens. Later its links. Usually innate immune system, broad molecular patterns, react with variety of pathogens. But once adaptive, it's much more specific. So I said earlier, pathogen-associated molecular patterns are the innate immune system recognizes. So these are from the pathogen. The pattern recognition receptors are from our body cells. So they are microbial cell wall component. Complement are the pattern recognition receptor here. So leads to opsonization, complement activation. Likewise, there are toll-like receptors. They react with these pathogen-associated molecular pattern. Lipoprotein of gram-positive bacteria is cell wall component. Leads to macrophage activation. These are in our neutrophil and macrophages. So with this is the signal one way. This is the receptor. Signal receptor binds and leads to cascadic activation. That's what it happens. Then what I said, pathogen is there. With the presence of their pathogen-associated molecular patterns, our pattern recognition receptors identify there as non-self. Then in a innate immune system, activation is there. Complement is part of humoral response of innate. Likewise. Thank you.